talk. Today we are very honored to welcome Professor Sarah Baatout to talk about radiation risks in human space exploration. Professor Baatout is the director of the Radiation Biology Unit at the Belgian Nuclear Research Centre in Belgium. She is also guest professor at Ghent University and KULeuven, teaching and directing research in the field of radiation biology, radiation protection, space biology, and medicine. Professor Baatout, thank you very much for joining us today. The floor is yours.
Okay, thank you very much. Thank you very much for this invitation, and it's a real pleasure and an honor for me to join this event. And I'm really looking forward to the questions that I hope will come from the audience. So first, let me share my presentation. I have prepared some slides for you. I don't know if you can see my slides in full screen.
Yeah, we can see them in full screen.
Thank you very much. Thank you, Mathilde. So I'm going to speak today about radiation risks and human space exploration. And it's really an exciting time we are living right now. We have just celebrated 2 months ago the 60 years of the first orbit around the Earth with Yuri Gagarin on the 12th of April, 1961. And since then, a lot has been accomplished in terms of human space exploration. And the duration of missions have also tremendously increased from a few days, first a few hours, then a few days, to nowadays on board of the ISS, on board of the International Space Station, a duration of space flight that can be up to 1 year, so 6 months to 1 year, which is the typical length of ISS missions for the moment. And what is in front of us is really very exciting, because we are preparing future moon missions and missions to Mars. So the objective is not to stay for a few months anymore, but really to have space flights that can last for sometimes 2 to 3 years in total. And so we've got to really make sure that astronauts remain and are in good health and have a productive mission. So we have, since the first space missions, gathered a lot of important data and information about how the human body reacts and responds to the space conditions. And we will see, in fact, in this presentation, how much this response varies between astronauts, and how we are preparing also right now what we call precision medicine in astronauts. So we are preparing missions that are going to last even longer than 6 months, 1 year. And this big endeavor is called the Deep Space Getaway. This Deep Space Getaway is really like the first human spaceship to explore the solar system. First, as I mentioned, around the moon and on the moon, and there later on future missions are foreseen to go to Mars. So it will be the Deep Space Getaway is the most distant human space missions that have been ever attempted, and it's also the testing ground for challenges of long duration human missions in deep space. And we will speak also a lot today about one of the main showstoppers for these missions, and it's ionizing radiation. So we'll focus on that a little bit more later on. So nowadays, a lot of the research is taking place for the moment on the International Space Station, as you can see on the left-hand side of this picture. And I will give you some updates about the type of experiments that are taking place, and some of our experiments that are taking place right now on the International Space Station. It's really the place to start to develop all kinds of technologies, all kinds of experiments that can later on maybe fly to the moon and or to Mars later on. So basically, in the 2020s, the idea is really to develop missions in the lunar vicinity, really around the moon, in a cislunar activities, and then hopefully on the moon with the development of a moon village. And after 2030s, leaving the Earth-Moon system and reaching the Mars orbit. And for that, we need a lot of disciplines. The space field is an incredibly multidisciplinary field that really needs and requires a lot of cooperation and collaboration between different types of expertise. And I guess some of you have already understood that from your discipline and in your interest related to the space sector. So the moon program, first let me focus a little bit on the moon program, is called the Artemis program to kind of counterbalance a little bit the Apollo program. That was the program to the moon in the 60s and 70s. And in the Apollo program, you remember 1962, the President Kennedy made his famous speech, gathering and stimulating the whole space sector to go to and to reach the moon by the end of the decade. And in 1969, the first Apollo landing happens on the moon. At that time, the NASA budget was about 4% of the federal budget, whilst nowadays, the NASA budget is about less than 0.5%. So the NASA Artemis program, and the Artemis program is not just NASA, it's also a very international program. We will see also that Europe is very much involved in that. The plan is to land the next woman and the next man on the moon around 2024. The first experiments are being developed very, very actively. And the goal is really to establish a sustainable presence on the moon to support, to test any type of future missions to Mars. So we are not, it's not an only a NASA endeavor, it's really an international endeavor. And I just want to kind of give here a short update about what is going on. We see that the ISS is really one of the main places for research and for activities, not only by the American side, but also by the Russian side, by Japanese, as well as Canada and Europe. Europe is extremely active in that. I will focus on Europe just in the next slide. But we are also preparing another and further future missions, so beyond low Earth orbit, with the Gateway, as I mentioned, where we also see that a lot is being developed in terms of experiences and a lot of technologies. We see that China is moving very fast, in an incredibly fast and incredible speed. Today, for example, the second part of the space station, of the China space station, has taken off with some future astronauts that are going to be in space. And in this context, my institute, the Belgian Nuclear Research Centre, has submitted 2 years ago, together with different groups, and coordination is taken care by Norway. We have submitted an experiment called Tumors in Space to fly to the China space station, hopefully in 2 years' time. And the idea is really to understand how tumor cells behave in terms of 3D models in space conditions. And can we make better models of tumors in space conditions? So as you can see, this is what is written in red. But not only the US, China, Russia, Japan, Canada, Europe, but also India is very much developing the access to low Earth orbit. The United Arab Emirates also very much pushing the space sector forward with their first ISS astronaut flight with some probes around Mars. So it's very booming. And Israel as well, with a first experiment that on the moon, and a second experiment that will take off very soon. So if we look now, what is the interest of the different and the main powers in the world, we see that beyond LEO, we have the US mainly playing an important role, China, Russia, Japan, and Canada. And then for the Moon and Mars, we see that again, it's a very international endeavor. So with a lot taking place and a lot of future technologies and technologies that are taking place. Now, let me focus a little bit on the European point of view. And ESA, the European Space Agency, divides human space exploration into 4 different cornerstones, so 4 different pillars. And the first pillar is what we call humans in low Earth orbit. So this is humans in low Earth orbit, in LEO. This is what is taking place right now on the ISS. So this is one of the programs from ESA. The second program is humans beyond low Earth orbit. So this is the preparation of the Gateway, of Orion, the preparation of the cislunar activities around the Moon. And then the third cornerstone is robotic lunar exploration. It's really going on the Moon, having a kind of moon village, and developing all the technologies, the life support, amongst others, life support technologies in order to survive and have a productive mission. And then the fourth cornerstone is robotic Mars exploration. And this is really the way to move forward towards a future Mars mission. If you look on board of the ISS, for the moment, we have quite a few experiments that are taking place. We have some human research taking place, and I put in red where my institute, SCK CEN, is involved. And right now we are following up astronauts and cosmonauts that are spending between 6 months and 1 year on board of the ISS, and we are performing different types of analysis that I will explain more in detail later on. On board of the ISS, there are also some fluid physics experiments, some education experiments that are taking place, some biology experiments. I mentioned here the BioRock experiment, which is an experiment that is really the first step of lunar agriculture. It's really understanding how bacteria can extract from moon analog different types of minerals in order to stimulate the proliferation of plants later on. We also have an experiment that just came back on rotifers, on extremophiles, so microorganisms, organisms that are really able to sustain very high level of radiation for a certain period of time. And that's really important for us to understand what are the mechanisms that these microorganisms have developed, and can we use these kind of molecular mechanisms in order to sustain, to develop radiation resistance in astronauts. Then we also have on board continuously some dosimeters. Dosimeters allows us to characterize physically the doses of radiation that penetrate inside the ISS and also that reach outside the ISS. And so these doses are very important to monitor because they help us to model and to extrapolate what to expect for longer mission to the Moon. So SCK is also very much involved in these dosimetry applications and measuring continuously doses on board of the ISS. If you look at the science program from the European Space Agency called the SCI-Space program, you can see in a certain way with these blocks, basically the type of experiments that are taking place and the type of models that are used to simulate somehow space conditions. So we have the parabolic flights, we have the bed rest studies, I will explain that more into detail. We have the Concordia platform in Antarctica. We have the ISS, of course, with some biology, some astrobiology. We have the ISS with some human research on astronauts themselves. And we have some physics experiments, material sciences, soft and complex matters, some microgravity application programs. And then we have also some ground-based radiation platform that I will explain a little bit more into detail, and other ground-based platform. So in red, you see all the type of platforms that in which my institute is very much involved and where we are carrying out different types of experiments. This is a slide that I would like to present in terms of the type of risks that are really on the agenda in order to find ways to mitigate them before we go forward to longer missions to space. And these risks are based on a multilateral human research panel for exploration, the MHRPE. And they basically rank and make a rating of the different types of risks. And you can see on the left-hand side, on the left column, the type of risks that are the main ones for human space explorations that can be really hampering, inhibiting the missions, the future missions. And you can see that it goes from the musculoskeletal system, the sensory motor, the behavioral health and performance, but also the ocular syndrome, nutrition, dust exposure, environmental exposure. And you see the last line is really related to radiation, which shows a long-term risks of carcinogenesis, so higher risk of developing cancer due to radiation exposure. And so it's important, as you can see on the right-hand side of this figure, for the ISS, on board of the ISS, this radiation risk is not really that important. And we'll see that there is a big difference between radiation on the ISS, on the International Space Station, and radiation to go to Mars. We are speaking about different types of doses and quality of radiation, so and longer missions. So we'll see that the risk is much higher. What is important on this picture is that basically to go to Mars, which is the last column, you see that you have quite a lot of red squares, and these red squares means that exposure for a longer time, so missions to Mars will induce longer risks and will require mitigation, so developing ways and countermeasures in order to avoid these stresses and these risks. So space radiation is the #1 risk to astronaut health beyond low Earth orbit. So in low Earth orbit, we have mainly electrons, and we have also quite a lot of protons that are irradiating. And we will see also in terms of doses what it corresponds to. But space radiation beyond low Earth orbit to go to the moon, to Mars, will be really a showstopper, because we have then 3 kind of components to take into account, with 2 of them that can be quite problematic. First, we have the solar wind. The solar wind is composed of low energy electrons, protons, and alpha particles. It's a continuous flux of radiation, but it's a low hazard. It's not something we are very concerned about in terms of radiation protection. However, what we are more concerned about are solar particle events. It's a flux, a very important flux and very dense flux of high energy protons. These solar particle events are not constant, they are intermittent, so they can last for a day or 2, and we will see on the ISS what is the place where the astronauts are going to kind of shelter in case of this type of events. But they can be very hazardous, so it's very important to protect against them. And there are different ways to kind of protect them, and we will speak a bit more about that in the future in the next slides. And then another main concern for us are galactic cosmic radiation with high energy protons, mainly alpha particles that can really irradiate with very high damage, also heavy ions. And it's a continuous flux. So basically, it's almost impossible to get rid of that. And they are very hazardous. They are hazardous. We need to really continue to develop ways in terms of shielding, but other ways also in terms of pharmacology, in terms of genetics, biology, regenerative medicine, in order to counteract and mitigate these effects. Then on top of the 3 types of radiation I've just mentioned, we also need to take into account the secondary radiation. And the secondary radiation come, in fact, from the first irradiation that I mentioned, and the interaction with this first irradiation with the material of the spacecraft. so the coating of the spacecraft, but also can interact inside the human body. And this secondary radiation can be even more harmful than the primary irradiation, and because they can really lead to the production of secondary particles that can be quite detrimental for human health. So we've got to be very cautious in the type of space material that we will use for space habitat on the surface of the moon, but also for space ship to go to the moon and to deep space. Here I mentioned to you that on board of the ISS, we have 3 places for the moment that are very well protected, where astronauts can shelter in case of solar particle events. And you see in the middle the water wall, which is basically the container that contain the water. So that's the water container. Water is composed of atoms from hydrogen and oxygen. And the hydrogen is a very small atom, and it's very good to protect, to stop basically irradiation and cosmic radiation. So it's a very good and a very good protection. Then you have the lab, the sleep station. The sleep station is also quite well protected with polyethylene. Polyethylene is basically the plastic that we use. Plastic bottles are composed of polyethylene. These are long molecules of carbons, and on each side of the carbons you have hydrogen. So the hydrogen atoms are really blocking and stopping the cosmic radiation, solar particle events. And on board of the ISS you have different types of alarms that are helping the astronauts and telling the astronauts and guiding them to shelter in these places where they can stay until it's safe again on board of the ISS. So what type of radiation doses are we talking about? First, we have to mention that it's why we need really well to characterize the doses on board of the ISS, because each mission is different. Radiation doses are mission specific. They will depend on the destination. So we have different types of radiation in low Earth orbit in comparison with going to the moon or Mars. Depending on the duration, depending on the vehicle, the material of the spacecraft, we have seen that the production of secondary particle can be quite important. So depending also on the habitat design, and also depending on the solar conditions. So all that will makes that each mission is specific in terms of radiation doses. But roughly, if we have to give some kind of a rough idea of the dose that the astronauts are encountering on the International Space Station, we have about 150 times higher doses roughly than on Earth. So on Earth we have about 1 millisievert per year as natural exposure from the outside environment, from the sun, from a little bit, a very small bit of cosmic galactic radiation, but also from the Earth radiation from the planet itself. For lunar missions, so if you imagine that, for example, you have an EVA, what we call an extravehicular activity. So you imagine we have to take that into account, that on the moon village, you will have an exploration of the astronauts. They will go and visit and explore and take some samples on the surface of the moon. And so they will do some extravehicular activity. So going outside the shelter of the moon habitat and then explore. So the spacesuit to kind of explore the surface of the moon will be their only protection. And we'll speak a little bit about the spacesuit in a moment. Because it needs specific protection in order to allow in a safe trips, because it's supposed to be used also for a certain number of times. The astronauts are going to do quite a few exploratory missions on the surface of the moon. And then if you count a year stay on a lunar base, so you arrive at about 300 to 400 times higher doses of irradiation than what we encounter on Earth. Then for deep space journey, leaving the Earth, going to the Moon, going to Mars, you arrive for a 1-year mission, going to Mars mainly, to 500 to 600 millisieverts. So we are now at about 500 to 600 times higher doses than we have on Earth. And then a full trip to Mars, going to Mars, staying on Mars, going back from Mars, so about 3-year trip. We speak about 1,000 to 1,300 times more radiation than we are encountering on Earth. So this is really quite important dose of irradiation. So to develop all these, the know-how, the necessary know-how and expertise in view of this future Mars mission, the ISS, the International Space Station, is really critical to test, to mitigate different types of technologies to decrease as much as possible health and performance risks. So the ISS, a beautiful picture of the ISS, is in orbit, 300 to 400 kilometers around the Earth. In orbit, it does an orbit of every 1.5 hours, so 16 times per day the ISS is going around the Earth. And the dose of radiation is about 150 times more than on Earth. So right now on the ISS, my institute, the SCK CEN, has got some radiation detectors inside, outside the ISS. We are studying the health impact on the astronauts, on cosmonauts and astronauts. We also have a bioreactor that has been flying in order to start this development of life support systems, so being able to regenerate as much as possible by the use of bacteria and plants organic matters, to recycle urine, to recycle feces. And we also had this lunar agriculture experiment, BioRock, that flew not a long time ago. But then, as mentioned, to go to the Moon, we are then encountering 300 to 400 times higher dose of irradiation. The Moon is very important for us because it's the archive of Earth's early history. There is no magnetosphere, no general atmosphere. There is so no protection from harmful space radiation. And it's the springboard and the testing platform before going to Mars. And the idea is really to develop a moon village that will be the first step before Mars. And on the Moon, as mentioned, the spacesuit will be crucial. What do we know from the Apollo mission? Less than 20 extravehicular activities have been taking place. So 20 times when the astronauts really went outside the spacecraft and on the Moon during the entire Apollo program. But the future lunar program are really imagined and developed in order to have about 70 lunar extravehicular activities during a 6-month period mission per astronaut. So it's not nothing. It's really, it requires a lot of development in order to have this perfect spacesuit, not only in terms of the design. We know that the current spacesuits from NASA are composed of about 12 layers. But it needs also to be resistant to abrasion, to reactive chemical dust, to UV, to temperature, to oxygen, to weight, but also against radiation. But it needs to be very flexible, elastic to allow walking. So different types of materials are being tested for that. And SCK CEN is also very much looking to the different ways of protecting the best with the best material for space use, but also monitoring radiation. And then to go to Mars, 1,000 times more irradiation than on Earth if you count the whole trip to Mars and back. Mars is the most similar planet to the Earth. It allows us to understand whether life existed elsewhere in the universe beyond Earth. It's really a fundamental question for humanity. We know that it has once been full with water and warmer, and it has also a small atmosphere of 1%. So it represents an habitable environment. The distance from the Earth to Mars is about 225 million kilometers. So at my institute, Belgian Nuclear Research Centre, we have a very strong program of radiation protection for human space exploration to support a productive, healthy human space exploration program of the solar system. And it mainly consists of three cornerstones. One is dosimetry, understanding and measuring, characterizing radiation inside, outside space vehicles. Another cornerstone is microbiology, developing life support systems in order to allow life to continue to develop, and astronauts to be fed, oxygen to be produced, thanks to the help and the support of bacteria and plants. And then the radiologists are studying the impact of radiation and cosmic radiation on the different parts of the body. So radiation monitoring, as you can see here, some in orange on the picture are some of the dosimeters, which is a collaboration between the German Space Agency and Belgian Nuclear Research Centre to understand the space radiation and how it penetrates basically inside the International Space Station walls. So for that you need you need active and passive radiation detectors to really map the radiation in all the modules inside the International Space Stations. And for that, we use, in the case of my institute, SCK CEN, we use passive detectors that record the total radiation that has been absorbed, and we change these detectors. So every 6 months, these detectors are coming back on Earth, and we send to the ISS fresh detectors and new detectors. But on top of that, we also need some active detectors. Active radiation detectors means that they can measure continuously and on the spot all the dose of radiation, and they can also measure the fluctuation in the radiation levels in order to create a complete mapping and a complete picture of space radiation on board of the ISS. Here is one of our national heroes here in Belgium. It's Frank de Winne, who was placing some of our detectors in the different places inside the Columbus module, inside the ISS. Another experiment are detectors that we have placed outside the Columbus module that you can see here on these nice pictures outside the module. And also very importantly, you need also to measure somehow what is penetrating inside the humans. And for that, phantoms are very, very useful and very important. So to measure the physical dose inside the phantoms helps us to understand what is going to really penetrate inside the human body. And for that, you need phantom measurements, and we participate to the Matryoshka famous experiments together with the German Space Agency in order to understand what is penetrating in terms of radiation inside the different parts of the body, but also to test different types of coating and shielding that the astronauts could wear. And you see on the left-hand side a vest that can be very useful for protecting against irradiation. Then this was for the physics part of the experiments that are taking place on board of the ISS by the dosimetrists of my institute. Then the microbiologists are mainly working basically on developing the Melissa loop. And the Melissa loop is a very efficient way to recycle organic matter. And it is composed of 4 different compartments. And each compartment is including very specific bacteria. And these bacteria will do their job by recycling partly and together, each of them with a specificity, different parts of the organic matter. Which means that you have the organic matter coming from the astronauts, you have a liquefaction, a carbon transformation, nitrogen transformation, and then you arrive at the compartment number 4 that produces food, water, and also oxygen by the use of bacteria and the use of plants. And all this goes back to the astronauts. So life support is extremely important for long-term missions to the Moon and Mars, and we are very actively working on that. So since the first experiment that flew in 2002 with the first mission of astronaut Frank de Winne, we had about 15 experiments that flew so far. So we have about 20 years of experience and unique track record of biological spaceflight experiments. And you can see some of the experiments on the right-hand side, from the very first experiments that flew with agar petri dishes to now very much more complex and active liquid systems where you have plunger, you can change the medium, you can refresh, you can do different types of activities of your cell culture system. This is a small illustration of our SpaceX experiment called BioRock, lunar agriculture, with some pictures of the rock and the bacteria that are basically extracting, purifying different types of minerals from these lunar rocks, and by purifying that, using that to for growth of plants later on. So these are the very first step of lunar agriculture that took place recently on board of the ISS, thanks to the help of SpaceX. And then in terms of radiation biology and space research at SCK CEN, it goes really in different types of programs. It goes from radiation biomarkers, understanding, developing the best biomarkers for radiation response of astronauts, being able to monitor the best way the damage inside the astronaut body, but also predict the risks that can happen from radiation exposure. Then we also have some tests on pharmacological tests in order to understand what is the stability of the pharmaca in the space environment. Is the life shelf the same, or is it decreasing? And how is it as efficient? We're also studying the impact of space conditions on the brain cells, on neuron network, on the microbiome, on cardiovascular risks, on the skin, and on the immune system. And all that is really to better assess the risks of radiation on a medical point of view, accidental exposure or space radiation exposure. So I would like to move forward now to what I call the precision medicine in astronauts, which is something where we see we really have to develop that much more, and we are currently doing that at the level of Europe. And there is currently a topical team at ESA really specialized on personalized medicine. And basically, what does it mean? It means that basically you have to develop ways to take each astronaut separately and as an individual, depending on his or her own characteristics. That can be genetic characteristics and sensitivity. First, an astronaut, we know that is exposed to different stresses, a combination of stresses. There are the physical stresses, like microgravity, like radiation, as we saw that radiation vary between mission, and like the microbes that are populating our gut. About 0.5 kilo of microbes are inside our guts. They are helping us to digest, to survive. And these microbes can be also impacted by space conditions. Some of the bacterial strains that we have in our guts might stop proliferating or decrease their proliferation, whilst others might increase their proliferation. So we have to be very aware and also conduct studies in order to understand what is the impact of space conditions on the gut microbiome. And then we have psychological stresses. The astronauts are subjected to very long isolation periods, confinement periods. These are bringing an increasing stress. They have also a very high workload, and they also can have some sleep disturbances. So what are the effects of space conditions on humans? These are variable between astronauts, so they will vary. They might not all have and experience these types of effects, but they are quite common to most of the astronauts. So you have the space sickness, the disorientation. You have also loss of muscle mass, loss of bone mass due to this microgravity environment, increased microbial virulence. We know that some bacteria can really start waking up, and viruses waking up as soon as the astronaut is in weightlessness condition, and that can be quite detrimental. We also have a disturbed sense of equilibrium. The movement is only recorded by eyes. Change of the liquid distribution with more liquid fluid in the upper part of the body, an increased risk of kidney stones, and disturbed, dysregulated lymphocyte function that decrease basically the efficacy of our immune system. But again, each astronaut will react differently, and we know that there are some differences between genders. So between female and male astronauts, we also know on an epidemiological point of view that there are differences, that females, for example, will have less hearing loss, will experience less hearing loss, will normally and so far are not reported to have any visual impairment in comparison with men astronauts. They also have a better immune response facing space conditions. But on the other hand, they score less well in terms of orthostatic intolerance, and this is how the way the cardiovascular system is going to react in space conditions. They also have a tendency to develop more urinary tract infections. And for males as well as for females, you also have a large variability in the muscle and bone loss. So there are some gender differences. Then, of course, the longer the mission and the higher the doses to go to the moon and to Mars, the higher the risk of developing cancer, because radiation can induce a higher level an induction of mutations, and we know that cancer is somehow also an accumulation of mutations in our genes. And we see that you have a big variability and variation in the risk. As you can see, in an ISS mission, the increased risk is not so big. It goes from 0.1 to 1%, 2% more of higher risk of developing a cancer. But when we go to and think about missions to the moon or Mars, we really are talking about risks to the moon that can go up to 10% higher risk, and even higher for Mars mission to about 30% in the extreme cases of higher risk of developing cancer. So, of course, these risks will vary between astronauts, but we see that we have to somehow take that into account in our precision medicine for astronauts. Also, not everyone is equal, so all humans are not equal in front of radiation. Some humans are more equal than others. In terms of sensitivity to radiation, you can see on this curve that some people, some patients, some astronauts also are quite radiation sensitive, as you can see here on the left-hand side of the curve. Whilst some patients, some people on Earth, some astronauts also are quite radiation resistant. And we can measure that, and it's what we do also on the astronauts that spend 6 months on the ISS. Before and after they leave, we monitor their radiation response and the damage that has been induced by this trip to the ISS. So it's important to develop biomarkers of radiation sensitivity and radiation susceptibility, and that to monitor basically and to understand which astronauts would be at higher risk. Also, radiation sensitivity varies with age. At very young age, of course, at the embryonic stage, the fetal stage, radiation can have detrimental effects, which also means that if we think about colonization of other planets and having pregnancies during on the moon, on Mars, the embryo, the fetus, will be extremely sensitive, and that can induce then some malformations in the embryo, or some lack of repair, and maybe some other effects later on of early development of cancer in this population. But radiation, the good news is that radiation sensitivity decreases with age. So you see that around 50 years old, you have much less radiation sensitivity. So you are more prone to tolerate a higher dose of radiation, as you can see on the right-hand side. This is some data from NASA, where you can see that basically the risk, if you count the gender and the age, that the risk is higher in females in comparison with males in terms of radiation, but that this risk decreases with age. So the higher the age, the lower the risk. Then to add to this complexity of differences depending on the age, depending on the gender, depending on the genetic background, we also have differences depending on the organs. And what we have a few organs that are very sensitive and are more sensitive than the rest of the body. And that's what we call the organs at risk from radiation. We have the eyes, we have the lungs, we have the breast, we have the stomach, the intestine, the bone marrow, and also the reproductive system. So these are the most sensitive, radiation-sensitive part of our body. So radiation risk, as you can see, will increase if you look at the ISS, the Moon, and Mars. We are really talking about to the Mars and back. To Mars and back, we are talking about 1,000 higher doses of irradiation in total, with higher risk, as you can see. So for that, it's very important to develop a roadmap, a strategy of research at the level of the European Space Agency. The latest one, which is published, dates from 2016, but at the end of last year, we've done with about 100 scientists all over Europe a tremendous exercise, together with ESA, of course, to now have a new roadmap, an updated roadmap for 2021, in order to really decide already for the next 5 years what are the main priorities in terms of research, in terms of what do we need to understand on a biological point of view and also on a physical point of view in order to prepare the future mission to the Moon and to Mars. So I would like to say a few words now about what a biomarker is for precision medicine in astronauts. A biomarker is any type of measurement, any type. It can be a protein, it can be a gene. It's any type of measurement that will reflect an interaction between a biological system and an environment. The environment can be the space condition, for example. This environment can be a combination of chemical, physical, or biological factors. And in terms of radiation response, we have 4 kinds of biomarkers. Biomarkers of exposure. So these are molecules, proteins, genes, different types of molecules that are basically going to be produced as soon as radiation exposure is taking place, and the expression will decrease with time. Then we have biomarkers of susceptibility. This allows us to, beforehand, before radiation exposure happens, to decipher which people are very sensitive to radiation. Then biomarkers of late effects. It's to predict very early, at a very early stage, what is the risk of developing a radiation diseases later on in life. And then biomarkers of persistent effects are biomarkers that allow us to measure at long term and that will stay almost permanent in the body. Here, again, you see in red the type of biomarkers in which my institute is involved. You have the epigenomic modifications, the transcriptional and translational modifications, the cytokines, the proteomic changes, and the DNA damage and the nucleotide pool damage. So what are the potential space radiation health risks? First, higher risks of developing cancer. So radiation carcinogenesis, space radiation exposure might increase cancer morbidity and cancer mortality risk in astronauts. Then we know that we might expect some acute and late effects in the central nervous system. Also, some cardiovascular diseases can be with a higher incidence, and other degenerative diseases. And the acute radiation syndrome, it's a very high response of the body following very high exposure for short term, for example, due to solar particle events. If you would have a solar particle event and you would be with a spacesuit which doesn't protect anything against radiation, which has no shielding, and that you would leave the astronauts for 2-3 days outside, you would really predict an acute radiation syndrome. And on top of so these effects, it's really important that there are different types of analogs. that allows to understand also parts of the space effects. And at my institute, we are using different types of analogs. We use the ground-based simulation. You see on the left-hand side, top left, a random positioning machine. We also have access to different types of radiation inside in Belgium, but also in the different installation and institutions like GSI or Ganil, GSI in Germany, Ganil in France, for example. We also go to South Africa or to Groningen in the Netherlands to irradiate with specific types of irradiation. We also have access to a microgravity platform. The bed rest study is a very interesting platform that we use also for many years, where we can simulate microgravity for long term. And then we also have access to different types of stations in Antarctica. You see a beautiful picture of the Concordia station, where we conduct research since 2010 continuously, where people basically stay in isolated and confined environment for a period of 12 months in a row. And we also conduct studies on the Princess Elizabeth station, which is the Belgian station in Antarctica, where I was myself happy to stay there for a few weeks 2 winters ago. So you see a picture on the bottom, middle bottom of this picture. Then we have parabolic flights that allows us to test different types of hardwares, and then we have different types of experiments that we run and that we perform on board of the ISS or other other spacecraft. So monitoring of radiation sensitivity in space, this is what we are currently doing for a few years now, where we monitor the blood parameters of astronauts and radiation susceptibility, radiation sensitivity before and after flight, in order to really understand how diverse, how variable this response to radiation is between astronauts, and in order to see whether there is a kind of common response and individual response. We're also conducting, that was also together with SpaceX, and this project was coordinated by Daniela Grimm in Germany and Denmark, understanding how blood vessels are developing in space conditions. Blood vessels are quite sensitive to space exposure and space conditions. So it's really important in order to avoid the risk of hemorrhage, to understand how we can better kind of protect these blood vessels. Another 2 experiments that we flew a few months ago and then last year also are rotifers. Rotifers are very resistant organisms that can survive in dry conditions, in a desiccated form that you see on the picture, but also in water. They can live in water condition. They are very resistant. Last week there was an experiment that was that started on tardigrades, and tardigrades are also very extreme organisms that can also sustain very high level of radiation. So these type of experiments are very important for us to understand what are the molecular mechanisms that these organisms have developed, that we could maybe develop, at least boost in humans in view of future exploration to the moon and to Mars. Then we're also studying the skin. Why the skin? Because it's the largest organ of the body. It has many different vital functions. Barrier, as an important barrier of our body, immune defense, it protects us. It's very important for thermoregulation, for sensory function, for endocrine function. And it's also one of the most frequent medical incidents that are reported, that is reported by astronauts, for example, from small injuries of the skin, or from dryness and pain from this dryness, or itching, or delayed wound healing. These are really very often reported by astronauts. We know that in space, skin is thinning. The epidermis is very much thinning, up to 20%. We also know that there are differences in the collagen to elastin ratio. There is also a loss of elasticity of the dermis. And there can be also an atrophy of the dermal matrix. And all these phenomena are also very closely related to skin aging. So it looks like really skin is accelerated, that the aging in skin in space is really accelerated. So we use mice for different types of studies in order to understand in fibroblasts, but also using hind limb models of microgravity, how we can mimic this type of effects. We also send some skin cells to space in order to understand basically what is the impact of space conditions. And you see one of our experiments there, where you see the culture of our space skin cells on the right-hand side, and the ground control. So the same type of cells that were developed and in cultures on Earth. And you see that the DNA damage, which is represented by the green and the red spots, is much higher in space conditions. We also studied the impact of space conditions on the brain cells, on neuronal network. And you can see some of our pictures here. On the right-hand side, top right, we have the random positioning machine. And the random positioning machine allows basically to have cells in floating condition continuously. And that allows us to really mimic microgravity weightlessness on cultures, on cells in culture. And you can see that basically what we observe is that on the left-hand side, you see the normal neurons, and then in the middle, neurons that are exposed to simulated microgravity, where you have a lot less proliferation, and even more when you see on the right-hand side with simulated cosmic radiation, where you have shorter neurons, shorter neurites of each neuron. So you see that you have a kind of an impact on the neuronal network in space conditions. So important to measure that. Also important to measure the gut microbiome, and we've published a few days ago the first studies on the change of the gut microbiome in mice following radiotherapeutical exposure. So really to understand what is happening when you have a pelvic irradiation, for example, in patients that have a cancer in the colon or in the prostate that are going to receive some radiotherapy treatment, what is the impact on the microbiome inside the gut? So we understand now much better what are the changes that we can expect in the bacterial composition. And it's also very important for the space environment because we really want to keep a healthy gut, a healthy microbiome with the right composition and the right bacteria in the right concentration. Very importantly, also, it's important to develop regenerative medicine, to really think outside the box, to be able to regenerate in case of need, not for tomorrow, but for the future, different types of organs in case of a massive dysfunction of organs in space. So we are working on that as well. As mentioned, we are also taking advantage of the beautiful programs, radiation research program from ESA. The IBER program, Investigation of Biological Effects of Radiation at GSI, is one of the programs that we use a lot to irradiate and to understand with different types of space simulated radiation what happens at the level of the cells, and in terms of dosimetry, what types of doses will happen inside the cells and how to mitigate these effects. These are pictures of the bed rest studies that are taking place in Germany, in France, where basically astronauts are placed in a position where you see that the head is lower than the legs, and that allows to simulate microgravity with the body fluid to the upper part of the body, and with that we can really conduct very important studies to understand what is the impact of simulated microgravity and developed mitigation ways, countermeasures, testing different pharmaca, different types of food supplements. This is a picture of the Belgian Princess Elizabeth Station in Antarctica. You see that it looks a little bit like a spaceship. In fact, it's very modern, very nice. Antarctica is one of the highest fidelity real-life analogs for future lunar and Martian habitats. So it's really an analog that we use a lot, especially the Concordia platform in Antarctica, but also the Princess Elizabeth Station, where we conducted different types of experiments on the immune system, on the heart monitoring, on the total full mapping of the dosimetry in Antarctica, and also on performance and psychological tests. We were also very happy to send this year some of our students. You see here 4 of our students on this picture that have been to the in simulated Mars analog condition for a week. This is the Analog Astronaut Training Center in Poland, and they conduct quite a lot of experiments that are space related, that allows us, for example, on the top right, you see different types of soil, lunar soils, Martian soils, and cultures of different types of plants that are taking place there. They do kinds of different types of other experiments in simulated microgravity. It's a very nice platform, and especially for students. So what are, that I already mentioned, there is a complexity in the biological effects of space radiation, because galactic cosmic rays are qualitatively different from what we know on Earth. They are more densely ionizing along the particle track. And you see on the right-hand side, basically, that they will really irradiate, ionizing radiation will really irradiate all along the track and will induce what we call complex DNA damage inside the cells that will make the cells much more fragile, take longer to repair the DNA damage. So galactic cosmic rays also cause unique damage to biomolecules, cells, and tissues, and they have a distinct patterns of DNA damage and repair length. So it's very important to understand that. They also have distinct biological effects and health risks. And right now we don't have a lot of data that exists to estimate the risk for heavy ions found in space. We also need animal and cellular models with simulated space radiation to gain new insight into the scientific knowledge. And we don't know if they will be a potential additive or synergistic modifiers, so effects of radiation combined with microgravity or psychological stress, for example. So we are looking at different ways of improving radiation resistance of astronauts, and that starts with shielding, of course, testing different ways of better shielding the spacecraft, but also the astronauts. But also medically understand the molecular mechanisms, and maybe select radiation-resistant individuals. Developing all kinds of radioprotectants that astronauts could take maybe just before they go out of the moon village, when they do their exploration on the moon surface, that they would take this kind of medicine in order to boost their immune system and protect them better against radiation. Different types of biomarkers that are developed in order to be able to monitor in each astronaut, depending on the radiation susceptibility of these astronauts, what is the best program of radiation, how many times the astronaut should go out of the moon base. Should each astronaut go the same number of times? So should we develop a specific program for each astronaut? And we are really going forward this way of thinking of having special program, individual program for each astronaut. Also developing regenerative technologies for different organs in order to be able to produce these organs on board. And let's not forget that the inspirational benefit of space. Space research is a very exciting field. It brings really a lot of positivity, and it's very well perceived by the public. It inspires not only the scientists, the the students, but also the very young children. We see that very often the primary students, secondary school students, but also university students, high school level. So it's really helping also to boost and to increase the number of STEM workforce. Let's not forget, we really need to have more scientists on board. And it brings easy and kind of positive communication and very exciting communication. So in conclusion, it's important to assess better, to continue this type of research on the impact of radiation for future deep space exploration. So it's important to have more insights into possible interaction effects of space stressors, to reveal altered cellular functioning, and to understand and to develop a possible countermeasure for future mission to Mars. If you are interested by the subject, I would like to invite you to read this novel, noReply, very interesting novel, which is a trilogy. So there are 3 novels, and in which basically I'm very honored to be one of the character, and I'm training astronauts that will go to Mars. So in the first novel, I'm training, and I'm the teacher for radiation protection. If you are interested by the subject, you can also look at the TEDx conference on that specific subject of radiation. I also invite you to read some of the references that are mentioned here. You can also, of course, contact me for more information. Let's not forget that human space exploration brings an awful lot of information and development for the benefits on Earth and for the benefits of treatment to our patients. So everything we do, developing different types of countermeasure, different ways of counteracting the impact of radiation, really helps a lot to better cure diseases on Earth. So there is really a very strong benefit for population on Earth. Tomorrow is an important day as well. It's the deadline for applying to become an astronaut. So I would like also to remind you to not forget this deadline. I hope a lot amongst you will apply before tomorrow night, midnight. I myself am finishing my application form, and I'm really excited to also apply for this. So please try it, and there is a place for everyone, and just would like to remind you about this ESA astronaut selection taking place for the moment. So with that, I would like to acknowledge some of my direct colleagues that you see on the left-hand side. It's a nice picture from my lab, and on the right-hand side, some of my collaborators, as well as the European Space Agency, BeSpace, and the International Polar Foundation. And of course, for anyone who is interested by this type of research, please join us, contact us. You are more than welcome. We have a lot of students. We have about at SCK CEN about 80 PhD students running in our institution, and we welcome about 300 master thesis students per year outside the COVID period, of course. So I hope that very soon we will be able to welcome again many master students and bachelor students. So with that, I would like to thank you very much for your attention, and I'm more than happy to answer to some of your questions.
Thank you very much, Professor. That was very interesting. So we had a lot of questions. The first one is why are vision issues different for low Earth orbits and lunar missions?
Yes, indeed. So basically, the risk is very different. For the moment, I would say, typically on the ISS, the doses of irradiation are and the exposure. Most of the astronauts, let's not forget, until a few years ago, European astronauts were not staying for such a long time on the ISS, and the typical missions were about 15 days. And then we went to longer and longer mission. And nowadays, the European astronauts are staying about 6 months, up to maximum 1 year. And we so right now, in terms of radiation exposure itself, the risk is still okay. It's still, we see some impact, some effects, differences between astronauts, but I would say that the major risk is coming from weightlessness, microgravity. There we see big differences between people. So with bone resorption, so osteoporosis. Osteoporosis is very much induced. For some astronauts, it's even 100 times faster. So 1 month on in weightlessness is equivalent to 100 months on Earth. So osteoporosis for some astronauts is very problematic, but not for all of them. And some astronauts are suffering from different things. Some of them have got some type of infection. We are particularly studying the immune system. Why? Because the immune system is dysregulated in microgravity. Dysregulated means that when it should react, it doesn't, and when it should not react, it reacts. So this regulation is quite bothering us, because when, for example, when the immune system should stay kind of quiet, not start to react, it can react. So it can, in astronauts, we are conducting studies, but also in Antarctica, about allergies, for example. This is typically when the immune system should not react and starts to react. Are allergies that we encountered, we have seen that more and more in very clean environment, like Antarctica, like on the Concordia platform, but we see that also in astronauts. Or typically, the immune system stops reacting, so you have a start of an infection, and instead of activating the immune system to start to fight against this infection, the immune system keeps quiet and doesn't activate. So this is also very worrying for long-term mission, of course. So the immune system is really something that is really important to tackle for long-term mission. Cardiovascular system as well. The cardiovascular system, we see differences. Basically, the cardiovascular system right now on low Earth orbit is the main concern, with higher risk of developing cardiovascular diseases. So all that helps us to understand, by extrapolating for longer mission to the Moon, to Mars, what could go wrong at a much higher intensity. And let's not forget that on low Earth orbit, astronauts are in direct contact with mission control on Earth. Basically, they can have an online consultation if something is going wrong. They can have an online telemedicine if they need to fix something. If they need to analyze an image from an equipment, a medical equipment, they can have the medical doctors helping them directly analyzing that. They have quite a lot of medical equipment, but for the Moon, to go to the Moon and to Mars, we will have certainly smaller spacecraft with a lot less material, lot less equipment, medical equipment. To go to Mars, no consultation, online consultation. So they will rely on themselves to operate, to do some surgery. On the ISS, if they need to come back, that can be, in case of really extreme health problem, it's possible to come back in a few hours' time. When you go to Mars, you have this issue you have to really sort out, and so that really requires a lot of even more challenges in terms of medical equipment, medical know-how, expertise, and things that can go wrong much more than on the ISS. So there are quite a lot of differences between the two that we need to take into account.
I really enjoyed your talk. I have a question. How have the MAVEN electric currents around Mars affected the preparations for protecting astronauts from radiation?
Yes, that's also a very good question. So that's something we are very much working on in terms of we have an ESA topical team especially devoted to radiation protection and radiation studies, and really to understand better what is happening, how we can protect. Should we have a habitat at the surface of Mars, or should we have something under the surface? How thick should be the layer of regolith regoliths. So how much underground should we go? So all these type of studies and modelization and extrapolations are really taking place in order to decrease as much as possible the risks for the astronauts. And it looks like, if I may answer to your question, I think it looks like the best way to protect from radiation would be certainly partly underground. But how much underground, this is still subject to discussion. How can which type and which depth of soil, of material, and maybe of water should we have? So all that is being really thoroughly studied, and certainly with the Mars missions that are taking place now and all the Mars measurements that are taking place now, it helps us also to understand what would be the best place also to have a Mars base in order to because we know that also the composition of the Mars planet varies, and there are places where you have potentially very interesting soil to work on, to use, and that could be quite fertile for being used with plants and so on. So that with lots of clay, for example. So this is really being studied very much by experts in the field, and I hope to be able to give you a very precise indication in a few years' time. Anyway, we will need a precise indication before we decide where exactly we will have the Mars base. But this is really being studied for the moment.
Another audience question. So since hydrogen is a good material to shield radiation, would it be interesting to use biomaterials to shield from radiation?
So it's a very interesting question as well. We thought, because indeed water is very interesting, so you would say, yes, in any cells, in any living organisms, you have a lot of water, so why not? And in fact, it's certainly not a bad idea. But then you need, the problem is that you would need to really shield properly, you would need about 10-11 meters of water or of living organisms, and 10-11 meters, it's really it's of thickness, it's really a lot. So water alone would not be possible. But I would say certainly if you can have if you can produce, if on Mars you could produce some water somehow, or extract water, and that may be something we can take into account. But let's not forget that every kilo we take on Earth to bring to the Moon and Mars cost a fortune, and is so we really have to we are really limited in the number of kilos we will take on board. So we really work on regenerative system, life support system, in order to regenerate from urine water, for example. But indeed, if in the ideal scenario, if we would have 11 meters of water, or simulated water with plants, with even using feces, could be also a thickness of feces, could in kind of really thinking outside the box could be a possibility of using this type of shielding as well. But realistically, it's not so. Not really so possible. But talking about biomaterial, it's you have very efficient fungi that you can use, that can reproduce very quickly, that could make with which you could make bricks. So in terms of building, maybe with biomaterial developing very quickly, making different shapes, the shape you want, you could imagine to have for the habitat. But in terms of shielding, we conducted some different types of studies, and especially with fungi, and it was not really efficient. It was not efficient at all. So it's certainly dry biomaterial. Bon, there is no water anymore in that. That was like zero in terms of radiation protection. So I would be not so tempted. But we conducted some studies with a space designer and different types of material, and that was not so good. So we just stop this biomaterial. But if in case you would have like 11 meters of these fungi living in water, developing very quickly, and being able to make some bricks for habitats and so on that could be potentially interesting.
Next question. Why are female astronauts more susceptible to orthostatic intolerance? That's
a very good question. We don't know. We don't know. We don't know. So why for orthostatic intolerance, we don't know why they are more sensitive. It's an excellent question. And we have studies on female astronauts, but we need more female astronauts and more data to really have also to see whether there is some individual susceptibilities. To do also, I would say what is really needed is some genetic studies between the different astronauts in order to see whether it's totally related to females or if there is an impact or combined impact of the age and gender. We don't know. That's a really good point. But so more studies are needed for that. We need more females in space. We need people to apply, more female to apply in the first instance. But very interesting question.
Thank you for that insight. What sort of experiments can be performed in Earth on medication to increase shelf life?
This is also a very important fundamental question. So in terms of pharmacology, for example, a typical simple example of an antibiotics. Antibiotics are used in space, but we know very well how an antibiotics will work on Earth in a patient. But in space, in microgravity, the molecular mechanisms of these antibiotics might be quite different. So the antibiotics might not be as efficient. You might need a higher concentration or a lower concentration of these antibiotics and might stimulate other molecular mechanisms. So there is really pharmacology is really a field where we need also more research. So if some of the audience, some people in the audience are interested in that, it's really an exciting field because molecular mechanisms of all the pharmaca of the space pharmaca that will be used need to be better studied. On mice, we have models on simulated microgravity. We have the hind limb model on mice. We use that also at SCK CEN. We can also test some pharmaca in the bed rest study. We can study them also in cells in the random positioning machine to understand if the molecular mechanisms are the same. And then what you can do also, so that these are biological experiments that you can do, but you can also do some pharmacological experiments studying the efficacy of a substance after irradiation. So you bombard them and you see how deteriorated they are. And if they age, if the medicine age faster. So it's also a question that we don't know exactly. We model, we know by studying some pharmaca on the ISS, we know what is the typical shelf life of this pharmaca, but there is really a lot more that needs to happen in order to study that much into detail. So there are other platforms also on the ISS where you can place, like I'm thinking about the ice cubes, for example. These are mini box of 10 by 10 centimeters where you can put different types of pharmaca, leave them on board of the ISS for, let's say, 6 months, 9 months, 1 year. They come back and then you study how did they behave in comparison with the ones that you kept in control condition on Earth, and see whether they lost their power, the efficiency, and then you can test them on cells and test them on animals in order to see whether they have the same quality. But we also when I went to Antarctica to the Princess Elizabeth Station, we also took with us 20 different pharmaca that are used for the moment, that are used on the ISS, in order to expose them on the roof of the station to the continuous UV. So the idea was really, because it was during the summer time there, so it's 24 hours of summer, 24 hours of light. So they were continuously exposed to UV, and that helped us for a certain number of weeks. And that helped us to understand how they degrade and how much faster they degrade than in a normal protected from radiation conditions. But a lot more needs to happen. And anyone interested, please, you can contact me. I'm really open to discuss that further, and but stimulate you also to also investigate it from your side.
I have a question here. It says, certain species of tardigrades are known to be 100 times more radiation resistant than humans through the action of the D-sub protein. Is there a similar mechanism known in the rotifers that you mentioned earlier? And further, is there a prospect of the proposed protection mechanism provided by this protein going to be deliverable to astronauts?
Yeah, excellent question. So in fact, it's very, very. It seems a simple question, but it's very complex, in fact, to answer to, because each of these extremophiles, the tardigrades, the rotifers, or bacteria that are extremely resistant towards radiation, some mechanisms are a bit common. In the way they repair DNA damage, the speed that they repair the DNA damage, the production of antioxidants, proteins, intrinsic production. This is kind of similar, but on the other way, some have developed specific mechanisms that we are understanding now more and more. So, for example, for the rotifers, we know that it's not only the DNA repair, that they are very fast at developing, at repairing, at detecting DNA damage. So one of the first things when you have the DNA molecule is when it's irradiated, the DNA molecule is in a natural state very, very condensed. The DNA is very condensed, and when it's damaged, you have this the fireman proteins that are going to induce the repair. They are big proteins, and to start the repair, the DNA needs to decondense. So needs to open up in a way to allow these proteins to start the repair. So if you decondense very fast, and some of these extremophiles are very fast at decondensing, then the DNA repair can take place very quickly. So we see that in some extremophiles that takes very quickly. Then if you have 200-300 times more of these proteins than in humans, of course, then it will be much faster, because you have these proteins that can go everywhere where you have the damage in the DNA. So this is one way they also have developed. Another way is that they are usually very much less conservative than human cells. Human cells, you have double checking that the DNA damage is well repaired before the cell goes to mitosis. And you have really a lot of cell cycle checkup that takes place. And if a cell has not repaired well the DNA damage, the cell will not proceed to mitosis. So you have really a lot of checkpoints that are taking place. And but in some of these extremophiles, there is no checkpoint. There is no, so they just continue. And okay, they will lose some cells. And you see that certainly in bacteria, but some will resist and will continue to proliferate. So it really depends on the organisms. In some cases, even if you only have one cells that continue and survive and 99 that dies, the cell that will continue to survive, the bacteria that will continue to survive and develop very quickly at a very high speed, and so will populate very quickly the bacteria that just died. So each bacteria and each extremophile has developed, I would say, common but also very specific mechanisms, and very interesting. So if anyone is interested in that, also contact me, because we're also working on these extremophiles, very interested in understanding better. So any help, any interest is more than welcome.
Thank you. So someone asked if we ever got the chance to send a satellite which would carry experiments into the radiation belt. What experiments do you think we should send to getting more insights into the risks of radiation?
Yeah, very interesting as well. All kinds of experiments. You can go from physics science, different types of dosimeters to measure the dose. I would send all types of shielding, different material. I would test different types of material and see what are the doses that are penetrating. I would definitely test also some regolite, maybe some lunar analogs, some Martian analogs. Different types of metals and aluminium, of course some lead, but different types of material or combinations of material to see which one would be the best shielding. I would also run some experiments with different types of extremophiles, different types of bacteria, different types of yeast, of fungi, lichens, also molecules of DNA, also look at how proteins would react. So I would send really a lot of different types of maybe these tardigrades, also other rotifers, all types of organisms in order to better understand. I would also try to understand the impact on electronics. I think this is also because what we want to avoid first, the impact on human health, that the astronauts are starting to get sick from the moment they are in space, and that we can't basically solve, and that they get sicker and sicker, and that they arrive half dead on Mars. That's not the idea. So we want them to stay really healthy and productive. But we have to make sure that all the equipment is going to work well, and that you are not going to have electronic failure. We see that very often when we do material studies and resistance to radiation, that you only need one very sensitive part of a whole equipment to break, and then to make it very complicated to fix so I would also test different types of material and electronic equipment to see what is their radiation resistance, because we need things that are going to stay and work well and for a long period of time. So we need radiation resistant material, and there we really need a lot of still a lot of research.
Another question, seeing as skin can be, I think you said, up to 20% thinner when they're exposed to radiation, how do astronauts protect themselves from this? How different is astronauts' sunscreen in comparison to regular sunscreen that we wear on Earth?
They don't wear sunscreens. They wear any kind of gel product. There are not so many possibilities that you can send up there in terms of product. So it's very limited. And you also have to, something I forgot to mention is that the way the heat is produced by the body, but the heat, because of the way the heat is distributed around the body on Earth, the heat goes away very quickly. But in space, the heat stays really stuck all around the body. So all the perspiration can be very uncomfortable for astronauts. So and it makes it also very with higher humidity at the level of the skin, because this heat stays and so you have also a difference in the skin flora, in the bacteria that you have at the level of the skin. It changes as well, because the humidity is also really. It's very paradoxical. Humidity inside the ISS it's very dry environment, but at the level of the skin, because of the heat convection and is very different. So you usually have much more humidity at the level of the skin. Not all kinds of product can be tested. There are some no sunscreen, definitely not, and very few cream are allowed. And a lot of research needs to also take place in order to make sure that they are still that the skin stays hydrated the right way. But what is mainly a problem is the wound healing. So astronauts, for example, one of the Belgian mission, an astronaut was saying that he cut himself at the very start of the mission, and it was a 15 days mission. He cut with an aluminium foil that was just floating in space and just cut his skin. And he said that for 15 days, the wound wouldn't heal. So the wound would stay open not move, so very sharp cut. So the astronauts disinfected it, kept some sterile gas on top of it for the 2 weeks. And as soon as he was back on Earth, the wound started to heal very quickly in a few days' time. But for the 2 weeks in space, nothing changed. So the healing was totally blocked, stopped. So there is really, it's very important. We for the moment, we have a student experiment called Forte, and the idea is really to understand. It's 3 students, extremely dynamic, 2 PhD students and a master students, and their experiment has been selected by ESA. And the idea is really to understand how wound healing, how fast it happens and it occurs in space conditions. Because if a small wound, doesn't heal, this is really an opening for infection, because any bacteria could enter, and because of the immune dysregulation and the fact that the immune system doesn't do what it's supposed to do and activate when it's supposed to activate, so that can really be detrimental. So I would say more than cream, the main issue is really the healing process, which is for which we really need even more research to stimulate wound to heal.
Thank you. That was really interesting. I think because it's almost the end time, we'll just ask one more question about you just talked about your students. Do you have any advice about to students in Belgians who want to go into that field or the space field in general?
I would say look around, contact people. Don't be afraid of contacting anyone. If there is a research that is particularly interesting to you, just look on the literature. Who are the specialists in every countries, and contact them and hassle them. And I think we are looking for, and I'm not the only one. Any research centers doing space research is really always looking for bright students and people who are interested in the field. And so for the moment, I've got students who are really in different fields. I've got 2 lawyers. One lawyer is studying the spacesuit and all the legal aspects and intellectual property of spacesuit. The other lawyer is looking at all the radiation protection of astronauts, and that's quite different than on Earth. So focusing on that. I've got a medical doctor looking at cancer, tumors in space and cancer problematic. I've got a physiotherapist looking at the cardiovascular system. I've got a designer who has just finished a habitat, taking into account radiation protection issues. But so I mean, the field is really vast and very wide. And so I would invite if you have ideas, contact, and not only when for master thesis or bachelor thesis, but also for internship. Holidays are starting very soon. Some of you have plenty of time. Maybe you can conduct some literature studies. Maybe there are really a lot of things to do. There is certainly enough. So please contact the people, the specialists in the field. If you don't know any specialist, please contact me, and I can certainly advise you in the different field of human space exploration, if it's the field you are particularly interested in. But there is certainly also, since yesterday, the young trainee program open at ESA. So I will send it to you, and you can send it to and post it. So you can apply. There are, I don't know how many positions at ESA for young trainees. So for people who have just finished their master or bachelor for a year, they can go for a year at ESA in one of the sites of ESA and have a fantastic experience there. So yes, please look at young trainee program of ESA. It just opened the last few days, and apply. And look at what is interesting you, but search on internet and contact people. Don't be afraid. And also there are a lot of conferences. There is also a Mars University taking place. I think they are still looking for students if they are interested. Look at Mars University. That's going to take place very soon, end of June, July. Every year we also developing our space summer school, but this year we couldn't organize it with the COVID, but we hope next year that we will welcome 20 students for this. There is also the FAIR. It's a radiation protection course, the FAIR course. That's for the people who are really interested mainly in radiation. It's open also up to the end. You can apply before the end of June. The FAIR, F-A-I-R. It's at GSI in Germany, taking place at GSI in Germany. Also 20 places are available there. Yeah, there are plenty of courses, yes.
Thank you so much for this advice and also for the fantastic talk. It was really interesting to hear about this aspect of space.
Thank you. Thank you so much for welcoming me so warmly, and good luck to all of you. And yeah, I wish you all the best.